From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on June 5th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 97th episode of Matters Microbial. As always, I am so very grateful for your listens and your views, and thank you for sharing the good microbial word with others. We are growing closer and closer to episode 100. It's kind of exciting for me. First, I received a comment I want to address briefly. Evolution is at the center of everything biological, and I sometimes describe it as the dead hand of Darwin upon all that lives. One listener or viewer objected to the term dead hand portion of that statement. Quite correct. Since evolution is an ongoing, living process, the statement should be the hand of Darwin is upon all that lives. Illustrated by this wonderful illustration by Rachel Webster. I just returned from a wonderful family trip, my spouse and our 2 sons, to Ireland, and Matters Microbial came with me. Here's an image of us at the Cliffs of Moher, the Cliffs of Insanity in The Princess Bride, and a relaxing moment at the Temple Bar in Dublin. It was a great time for all 4 of us. I continue to mull over new experiments and demonstrations for my fall course in microbiology. I need to remind myself, as in this image, it is not my first microbial rodeo, and I will keep you all posted. Here is a story that's relevant to today's topic. Many years ago, I noticed a microbiology student starting to look a bit distracted in class. So I pointed out to her that I was emitting a cloud of microbes as I spoke. She looked horrified and rushed home to create this image of Charles Schulz's character Pigpen surrounded by microbes. I treasure it, actually. I brought this topic up later in class and pointed out that we are breathing in one another's microbes. Katie McKissick, the wonderful Beatrice the biologist, created this amazing artwork in response. And there are actual research papers published on this very topic, which I will link to in the show notes. So we walk and we talk and we exist in a vast cloud of microbes. What do they do, for good or for ill? Thus, I am honored to introduce Dr. Ari Kozik of the University of Michigan to discuss the microbes all around us and how they interact with our very own airways. I also hope she'll tell us a bit about outreach and representation. Welcome to the Quality Quorum today, Ari.
Thanks. I'm happy to be here. Thanks for having me.
I am so pleased to meet you. I've followed your work for some time, and I was delighted to see in your emails that you included that wonderful quote by Zora Neale Hurston. Research is formalized curiosity. It is poking and prying with a purpose. I have that up in my lab.
That's great.
Well, I think you study something that's very important and very interesting. Now, you don't know this about me, but I was raised in the Los Angeles basin, and I was breathing a lot of junk all the time. And so the idea of what keeps our airways clean and clear is just one part of what you're talking about as well. And we tend to think about microbes here and microbes there, but you're one of the few people looking at airways. And I'm very interested in that.
Yeah. So, most people, when they hear about the human microbiome, they immediately think of the gut microbiome, I think, because it's been the most popular, I think, of the human habitats that we know that microbes reside. But until, I think it was 2010 or so, the general consensus was that, your lungs, your airways, your lungs are sterile. And if you're healthy, there weren't any microbes there. So if you opened a medical textbook, that's what they would, that's what you would find there. But, as often happens, technology kind of opens doors in lots of places. And so as we gained the ability to, take samples from different, body sites and figure out what microorganisms were there, we found that, actually there is a vast community of organisms in the lungs, just like there are in pretty much every other body surface that humans have. And so this is a relatively new, it's been about 15 years or so, relatively new field of research. But that's one of the, main reasons I was very drawn to it. After graduate school, I had worked on inflammation in the gut, and I was looking for kind of like a new area of microbiome research. And I was fascinated by learning about the airway microbiome and how understudied it was, and how we didn't know that it was, that it was there. So that's kind of how I got started there.
I do know that in the past, Ari, people had looked at, for example, the cystic fibrosis lung and the microbial populations and actual evolution that takes place within the biofilms in the lung. But this is different. This is in, quote, the healthy lung in some cases.
Yes, that's right. So, in cystic fibrosis, that's a disease where, a person's lungs become obstructed with this really sticky, very thick mucus that kind of acts as a glue and creates a different kind of environment inside of the airway that microbes can kind of thrive in. And so their microbial infections are a concern in cystic fibrosis, certainly, and have been. But the idea that, outside of situations like that and having, pneumonia or some sort of deep lung infection, the idea that there are microbes in our airways normally, and that's a good thing. That's kind of, this new idea. And so because of that, there's a lot of things that we have yet to learn about what those microbes are doing and how they are potentially helping us, and how our immune system, sees them, interacts with them, tolerates them, and then how that changes based on the environment that we find ourselves in. Because, I always like to say that humans are interesting because we are, we exist in an environment, but we are kind of our own environment in a way. And so it's really interesting to think about that connection between, where we are at any given time in or place, and how that's interacting with the inside of our bodies.
It's interesting in a way to me, Ari, and if I say something you don't agree with, please hammer on me about it. That's how I learn too. But when you're looking at the gut, you've got, and please forgive any jokes, I'm not trying to make any about this, the organisms that are just passing through, the ones that have co-evolved with the gut, and the ones that sometimes set up, I call them, forgive me, squatters, set up a temporary redoubt there. And it makes me wonder about the same thing in the lungs. Do you think there's evidence for a co-evolved group of organisms living in the lung?
So in my opinion, I think so. A lot of the organisms that we find in the airways, we actually think are seeded there by the microbial members of our mouth, actually. And so the upper respiratory tract. And so this is one of the places where it's really important, I think, to think about the different body structures that we have and think about them as potential environments. And so as you can see in the diagram of the respiratory tract, where we have, an upper respiratory tract, which is everything, from the neck up, where we have these spaces that microbes like to inhabit. And so that's, the nose and the sinuses, but there's a special area in the back of the throat where the esophagus, the pharynx, and the larynx all come together, where you have a like a toll booth almost, where particles can choose to go down the esophagus to the digestive tract, or into the larynx, down into the bronchi, into the respiratory side of the system. And these systems are kind of on top of each other in a way that has this epiglottis as the kind of control for kicking things one way or the other. And so because of that, our mouth is teeming with organisms. Our teeth, gums, deep in the tongue, there's lots of different surfaces inside of your mouth that microbes really like. There's, we're all constantly shoving liquids, biomolecules, foods, things there that they can feed on. So that's a very microbial rich compartment. But, as we breathe and, move about the day, we are having, microbes that are moving down from the mouth into not only towards the gastrointestinal system, but the rest of the lower respiratory tract as well. So that would be, your trachea, your primary like bronchi, and then deeper into the lungs, you have the alveoli and the branching that continues. And so we know from, studies where we've taken samples at these different places, or well, I should say other scientists have, and we know that, there is a microbial community that can set up shop in those different areas. But we noticed that that it looks as if there is this transmission from the oral cavity down into the airways. And this happens a lot, typically actually while people are sleeping. And so through this process called microaspiration. And microaspiration is where you have tiny droplets that kind of cross that, gastrointestinal to respiratory barrier. And so sometimes, you're laying down, things go down the wrong tube, things go down the wrong pipe sometimes. And so there's definitely ample opportunities for microbes to get from the mouth into either of those other two systems. And if you, think about just the mechanics of breathing, you're constantly, pulling air in, pushing air out. Whatever's in the air around you is going to be, sampling the air around us all the time. We can also think about, environmental features like humidity and temperature. It's kind of going to be closer to the, ambient temperature outside and in the mouth. But as you get deeper into the airway, things get a little warmer as you get closer to core temperature. And it's humid and, there's moisture. So there's like lots of things that can support microbial life. And so it's a really interesting and different niche for microbes than the gut, which is, like I said, what is typically what people are thinking about when they hear microbiome.
So how would you describe the difference in diversity, what you see with the airway microbiome to the gut microbiome, which everyone talks about?
Yeah, so the gut microbiome is much more diverse than the airway microbiome. There's, I don't have an exact number, but there's just lots and lots and lots of more different species in the gut than there are in the airway. According to, the data that we've collected, even in, in healthy individuals and in individuals with, asthma and COPD, we get, a handful of different bacterial genera represented. There are lots of different, species within these certain groups that are present, but it's nowhere near the same, expansive diversity that we see in the gut.
I have to tell you, in my mind, I'm thinking of all the artwork that I want to talk artists into doing about this, because it's 100% there. Are we talking aerosols, by the way?
Sure. I mean, in a way, yeah. I mean, I think everything that is generating droplets, the microbes can be in the droplets, which has become more of a, I guess, common awareness in the last several years now. But I think the droplets of, liquids, microbes, other volatile organic compounds, things that are around in the air.
I have something that I hope will make you chuckle. Again, hope. So as something I do with my first year students, I have them, please forgive me, swab their water bottles. I call them water bottle buddies. And we do 16S single colony on them just to see what's there. That's the only reason we're learning basic microbiology, isolating colonies, streaking them to singles, that kind of thing. What's interesting is I warned them, when you get 16S data from your water bottle buddy, don't be alarmed if the genus name is disturbing to you, because there are lots of different organisms all through you that have relatives that you've heard of that do bad things. And sure enough, I had a student who had Neisseria as the isolate, and all of the people in that lab started teasing her that her water bottle had an STD.
Oh, no.
And I had to keep saying, no, this is also, and I think I can use the word commensal for the Neisseria that live in the upper tract. And this is very similar to exactly what you're saying. When people use a water bottle, they're constantly aspirating and putting things around. It's the microbial world that we live in.
Mm-hmm. Yes, and I think that that's important to know also, because, one of the other areas of difference from the gut that we find in the airway is that, in the gut, there is this huge amount of taxonomic diversity. So by that, I mean, there's lots of different kinds of microbes some of them are, closely related to each other genetically, others are super far away. So we just have this really huge collection of organisms. But in the airway, what we see when we look at, healthy people is that it actually is dominated by a relatively small, handful of bacterial genera that are present. And so that's, for the most part, you're going to get your Streptococcus, Prevotella, Veillonella, Moraxella, Neisseria, as you mentioned, some Bacteroides, and Haemophilus. And in pretty much all of those genera, you're going to have what I call problematic species, and then some that seem to not be problematic. And we don't really know, what they're doing, but they're not known to cause infections or diseases themselves. And, my research is, focused around trying to understand more about those bacteria that we don't really know what they're doing. And I think that's an important step that I'm glad to see more people talking about in microbiology, because, I always like to tell my students that, bacteria can have impacts beyond just always good or always bad. And so I have, as you see in the other diagram, where I have this spectrum of activity, and, on one end we have, the good, or things that we think are beneficial, or, probiotics people might be familiar with, bacteria that seem to always be doing good things or being beneficial. And then on the other side, you have the pathogens, the things that make you sick, things that people will be concerned about, like Neisseria gonorrhea, for example. But outside of that, there's this real big space in the middle where you could have organisms that could be doing things that are positive, have positive outcomes, or not, or it could be influenced by the environment. It could be that they are, sensing different things. They're having different kinds of interactions. And we know a lot less about the microbes in that middle space than we do about the microbes that kind of exist on either side. And so I'm generally really interested in the microbes in that middle space to kind of understand what they're doing and how they support ongoing relationships between, our immune systems and the environments that we move through.
If you listen carefully, you can hear all of my microbiota applauding. And the reason I say this is that I had several discussions with Arturo Casadevall of Johns Hopkins. He doesn't even like the term pathogen. Now, I'm not going to go quite that far. Let's not redo the prokaryote wars. But what I will say is that he says that, for example, the E. coli in your gut that makes a particular vitamin, if it's released into your peritoneal cavity, can cause sepsis. So it's not the organism, but the relationship with the host. In other words, and it's a word you used in this diagram, context. And as a real estate agent might say, it's location, location, location. In this situation, it's context, context, context. And I'm just delighted to hear that the same kinds of paradigms we've seen in other niches are being followed with the airway, other than this very, not attenuated, that's not the word I'm looking for, restricted level of complexity that you see in the airway.
Yeah.
Has there been any work looking at the airway microbiome at birth? And, whether or not there are differences, whether you're born in kind of a hospital versus a non-hospital environment.
So for the infant microbiome, to my knowledge, I think most of the, airway or airway adjacent things you're going to find there are nasal swabs, actually. I think because of the accessibility, and, that's an important thing to consider when you're doing studies, especially with vulnerable populations, of which infants are certainly. And so there's a lot that we know about the community of the nasal compartment in very early life, people are looking at that in conjunction to what the gut looks like and the different environmental settings and exposures that kids can have, and how that can be associated with the development of wheeze, which is considered to be a precursor for the development of asthma as they get a little bit older into childhood, or the development of, allergies and things like that. And, kind of another factor that's kind of related to that is looking at the respiratory microbes in infants and how they interact with different viruses that they may be exposed to, like RSV, which is, really common in infants, or other kinds of, hospital acquired infections like that, and how that is interacting with the bacteria, and whether or not those bacteria can kind of help protect against the risk of some of those infections, or the consequences of those infections.
I think most people thinking about this, or listening to this podcast, or watching it, are wondering if there are differences in the airway microbiome that you would see for people in urban environments versus, say, agricultural environments, etc.
Yeah, so this is a really, really interesting area of research that I'm actually, really interested in, is because I think as I talked about the different kinds of exposures that we think about, in an urban environment, the kinds of things that you may encounter, whether that be smoke or industrial odors, or traffic, diesel exhaust, things like that. Just the general, soup, if you will, of what you're existing in is a little different depending on what type of environment you're living in. And so in urban environments, certainly there's going to be a different set of exposures than in rural environments. Now, in rural environments, especially if they are, agricultural, there'd be maybe more, plant related things, a lot more microbes that are from our, farm animal friends, for example, and how connected a person is with the outside environment. And those kind of microbes, we think, also may play a role in, shaping what your airway microbiome looks like, and how that interacts with your immune system. And we think that that's actually, one of the reasons why we find folks who live a more agrarian, like traditional, less modernized lifestyle, like the Amish, or like the Hutterite, for example, they have different rates of the development of asthma. And we think that part of that is because of the different microbial exposures that they're getting as they interact with their natural environment.
I have a question about possible interactions that take place. Now, I know that with Pseudomonas aeruginosa, the CFTR gene product is actually internalizes to get rid of them. And so there definitely are host proteins that are kind of trying to modify what's there. Are there other types of associations taking place with the lung? I like what you call it better than lung, the airway microbiome?
So I think so. I think that, and this is, speaking more directly to what the kinds of questions that we're asking in the lab, is that, because the source of microbes in the airway is so expansive, like the mouth, for example, as we talked about earlier, has like so much, there's so much there, there's so much coming in from the environment, and yet the airway seems to maintain this relatively stable community of a select group of organisms. It seems as though there's some type of crowd control going on. Where, you know, there are some bouncers somewhere, you got to have a VIP pass to get in. But we don't really know what exactly those mechanisms are yet. But I suspect that there are probably very important relationships between the host and certain of those microbes that kind of keep the different organisms that may come through that shouldn't really be there, that would be problematic if they set up shop, that kind of keeps them from doing that in, you know, the healthy context, whatever that means. I mean, obviously, we do have people you can get an infection where you have like an outgrowth of a particular, you know, organism or pathogen, but there does seem to be some balance or some process of keeping things, you know, hunky-dory in there.
Wonderful. So you're interested in this interplay, and you're kind of exploring the recesses of, I'm not going to call it microbial dark matter because that means something different to a bunch of people, but looking at the relationships between the organisms that exist there.
Yes.
And I think that that's very fascinating work. Why should listeners and viewers care about what we're looking for in the lungs?
I think there are a few reasons. One is because we are starting to see patterns where, you know, the airway microbiome seems to be linked in a lot of different ways to chronic respiratory conditions that have a really high burden in the US, but also globally. And so one of those that I study is asthma. And chances are everyone knows somebody who is affected by asthma or has been at some point in time. And asthma is actually one of the most prevalent non-infectious diseases that we know of. And it can manifest a lot of different ways in any different time of life. Most people are familiar with asthma that, you know, happens in children. It kind of develops early. Usually those children also have allergies or eczema or like inflammatory, like allergic skin reactions. They kind of go together as a package deal sometimes. And so people are generally familiar with that manifestation of asthma, but there's also, you know, asthma that shows up later. There's asthma that shows up after menopause. There's asthma that shows up that seems to be related to certain exposures, and that can happen at any point in life. And there's all these different patterns that we see that seem to be associated with these different groups of microbes or bacteria that we see in the airway. And so one of the things that I'm interested in is understanding how the communication between those microbes and our immune systems could be different, and, you know, kind of modulating how people are developing or experiencing their asthma, and whether or not that disease is actually responsive to the current methods that we have to try to help people feel better when they have asthma.
You know, I'm delighted delighted. I'm pleased to hear this because growing up, I had a terrible time with asthma because I grew up in Southern California before the air was cleaned up. Keep in mind, it's still quite dirty. Mm-hmm. And my parents were heavy smokers. Mm. And in fact, it was the asthma that my older brother and I suffered that finally got my mother to quit.
Yeah, because we were. Yeah, I was just going to say that, like, in addition to, you know, the developmental or the origin of asthma that we don't really know what that is, the other aspect of why this is important is because we think that this is really important for exposures and how exposures to certain things environmentally can impact our health. And smoke is certainly, you know, a big, one of the big concerns, and has been, but is now, especially as we think about, you know, wildfires and like other kinds of smoke exposures or chemical exposures or industrial, you know, fumes and things. And so people, you know, all over the world, and certainly in different places in the US, kind of experience different loads of that kind of exposure. And so how is our, you know, microbes helping us mitigate that? Is that something they can do? You know, I'm really interested in this concept of microbes that can, because we know microbes have this really cool ability to break down things sometimes. Like, are there microbes that can break down these different things that we're breathing in? Can can that help us? Is it something that we can harness? Those kinds of questions. So I think it's important for both, the chronic disease side of things, but also the environmental exposure side of things.
It's funny, we talk about the microbiology of the built space, right? Well, how about this built space?
Right.
And I was thinking about it now. What I'm going to say now is very elementary, but I don't know if all listeners and viewers have heard of it. We have cilia in our lungs called a ciliary elevator to help remove all the gunk that we breathe in. For example, the huge numbers of rubber particles from tires that we apparently develop in our lungs, right? And so, as you say, it might be very interesting to learn if the organisms in those areas are taking advantage of that. And I want to say that's good.
Right.
But we shouldn't be breathing bad stuff anyway, right?
Right. Yeah. Yeah. And I mean, exploring, like, we know that, one of the things that microbes are really great at doing is adapting. Yes. And so, like, is there, a chronic exposure that can, trigger some sort of like process? The microbes can have abilities to break down certain things that we don't know about that kind of you can gain. Maybe they're trying to protect their niche, which is beneficial for both of us. So, those are really interesting questions that I think will only become more relevant as, we continue this civilization. So I think that's really important.
It's funny. I make a joke of this, that I've never done anything research-wise ever that's benefited the human condition. So I'm very interested in folks who do things that actually touch human beings. And you're talking about an example of that. It must be very wonderful for you and your research group to have they can actually tell all of their family members exactly why their research is important.
I hope they're excited about that. That's certainly something that, I'm excited about. And one of the things that kind of motivate me. And I think, my own curiosity of, trying to understand how the world works and, how things, interact and can influence, different outcomes, has always been something that captivated me, even as, a student, learning about all the areas of science that I could potentially participate in. And I think it's one of the reasons why microbiology was so cool, because it's, a whole different kingdom of organisms that can have like very direct and immediate, but also indirect and over time impacts on humans. And I tell, the students in my microbiology class, microbiology interacts with our life in so many ways. And so a lot of what we do is connected to microbes in some way. So that's one of the reasons why I think microbiology is cool.
So that business of the cartoon that a former student who's a wonderful dentist, my God, I'm old. It's been a while, but that wonderful illustration of pig pens surrounded by microbes. Mm-hmm. I mean, you could say that you don't like that because it kind of is a representation of dirt, but it is true that we're all constantly moving through this cloud of microbes at all times, and they were here first. Mm-hmm. I have this saying, first evolved, last extinct. And so I really appreciate this work. I do know, if you don't mind, that listeners and viewers would love to hear how you take samples, or is this going to be a little disturbing to learn?
I don't think it's disturbing. I don't consider myself to have necessarily a strong stomach, but I guess it could be said, working with gut microbes, I'm very grateful to now work with airway microbes because the sample material is a little different. So for taking samples to study respiratory samples, you can do it a couple of different ways. It's a kind of a spectrum of invasiveness. And so the least invasive way is taking either like oral swabs or saliva samples. So people can, just give us some spit and we can, process that. But if we want something deeper from, the airway or the lung, like deeper down, then the next, most accessible sample is what's called induced sputum, which is what I typically work with. And so that is essentially mucus that people cough up. And so there's a way that we can, encourage people to cough when they come into the clinic to give samples. And I think it's just, a saline solution, and it kind of can help get some of that mucus that's down there detached, and it can be expectorated. And so we take that and run to the lab, quickly before things die off, and try to process it. And so that is the most accessible way. The least accessible way, but I guess there's a lot of research benefit to these samples, is if we actually are able to get bronchoscopy samples. And so these are from procedures where, an actual brush or it's kind of like a vacuum, like water tube, like you would get at the dentist, where they can, put liquid in and suck it out really quickly. But you can actually go, I think, like through the nose and down into the lung, but there's a camera associated with that. And so it's much more invasive. You have to be sedated. You need like a lot of, really skilled, medical staff to kind of help you get that. And so people typically, especially if they're healthy, aren't really signing up to, undergo endoscopic procedures. And so typically we can get those samples if someone's already undergoing that, to see if there's something going on, they need to get checked out. Then we can get samples that way. But the nice thing scientifically about those samples is that we know exactly where they came from. As far as, the lung is a relatively large space. You have all those different, branches and bronchi and levels, and you have 2 different lobes. And so when you do those kinds of invasive procedures, you know exactly, where the location in the lung that came from. That's less, it's not really possible with induced sputum, because you're just coughing it up and it's just coming from somewhere. But we don't really know. But there have been studies where we have compared, samples that come from the mouth, from the sputum, and from the different airway compartments. And it seems as though sputum is a reasonable representation of what you will find, deeper in the lung. And so that's typically what is used because it's way more accessible.
It's interesting when you talk about this, because I know that there was a technique. I was taught to call it lavage, where they go and wash out the lungs when people would inhale really toxic things.
Yeah.
And so this isn't a subset of that procedure, it sounds like to me.
Yeah. So we can take lavage fluid, or bronchoalveolar lavage. So that's we can get that wash fluid, and there are certainly microbes in there. Or there's a tiny brush where they can actually scrub. It's kind of like a rough word, but it's not that abrasive, but it's just a way for them to collect, a little bit of tissue from the inside of the airway. And so we can take either of those kinds of deeper lung samples for microbiome work.
In a way, this is relevant to people who do gut microbiome studies. If they just look, we're talking gross, I apologize, at stool samples, that isn't necessarily the same thing as what's hanging out on the epithelia inside. And so what you just described is probably shows you the organisms that are adherent to that section of the lung tissue.
Right. Yeah. And I mean, there's a lot of different things that can affect, there is some, surfactant in the lung fluid. There can be mucus of different viscosities, depending on if you're healthy or if you have a condition, more that's like CF, then it'll be like a different kind of environment. So I think it's, again, context is everything. So understanding if you're working with a certain group or a certain, cohort of people that you're sampling from, it's important to know those kinds of parameters.
I did have a question for you. One of the things that I talk about are some of the deadly centrisms of microbiology, and one of them is called oxycentrism, because the way biology is generally taught has everything to do with oxygen, and that's the knee-jerk reaction that students have to things. And it looks to me from some of the figures that you shared with me that there are organisms in our lungs that actually don't care for oxygen.
Right. Yes. And so this is something that I am, actively working on. And I think that, most people, when I talk to them and tell them, like I'm working on the airway microbiome, and then they are aware of some microbes and they're like, wait, what are, anaerobes doing there? And I'm like, that's a great question. And so that's something that we are working on. I look forward to figuring that out and sharing. But I think that there's a lot of things that we can now study about how organisms are interacting with oxygen and other different types of environmental factors that have been really difficult to do historically. And I included, I think, in one of the figures, what we used to have to do when we wanted to study organisms that didn't have or that couldn't, survive in oxygenated environments. And this diagram is from, I think, Pasteur's device that he developed way back in the day that consisted of, this flask where you have the medium where you're going to culture your microbes, and you boil it to eliminate all of the oxygen that's dissolved. And then after that cools, you connect it to a bowl full of hot mercury, which is everyone's favorite thing to do, and then flush that with carbon dioxide. And so you can have this, inoculum space where you can grow these microbes and there's no oxygen there. And so thankfully, that's not what we have to do now when we want to work with organisms that don't really care for oxygen. So now we have these really cool anaerobic chambers, as you can see in the photo, where we can have a workspace, kind of like a biosafety cabinet in a way, but it's a contained environment where we can put in our own mix of gases to kind of create our own atmosphere that doesn't have oxygen. In my lab, I also have an atmosphere where I can control how much oxygen is in the atmosphere, so that we can kind of put microbes in that environment and see how they're behaving when we change those environmental conditions.
I think it's wonderful. And for listeners and viewers, I mean, there are many techniques to reduce the amount of oxygen. Most people of my particular age remember things like candle jars. And the thing is that doesn't really control the gas mixture there, and it doesn't destroy that much oxygen. There are also small chemical sachets you can put in sealed containers, but you can't really do anything with those organisms. You just look at them. So when I would grow up Bacteroides, I could do that in a bag where I have eliminated all of the oxygen, but I can't manipulate the plates. The glove boxes, you can. And there's a really, I'll see if I can find a photograph of where people up the pressure inside so the gloves invert and stick out, and then they run through giving high fives to the hood. And I don't know if you've done that, but that seems to be commonly done in anaerobic biology.
Yeah, so we are really excited to be able to have a lab where we can work with organisms that have all these different lifestyles to kind of understand to begin to understand what they may be up to in our airways and what that means for us.
I think that's very interesting, and I know how difficult that work is, but you're finding out cool stuff all the time. One thing I would just add is that a lot of people make assumptions, not you, I'm talking about lots of folks, about oxygen. Let's just say that I look at colonies on a petri dish. Inside the colony doesn't have a whole lot of oxygen present. And that was such a surprise to me so many years ago when I actually had an opportunity to use an oxygen probe, and saw as soon as that probe hit the surface of the colony, the oxygen levels went way down. And so these are things that folks don't think about, and it brings to mind the question how you have anaerobes in the lung, or probably I should call them microaerophilic organisms that like a little bit, but not too much.
Yeah, and I think that, knowing or understanding how microbes are forming communities and what those communities, communities are structured, I think is something that will be important. I mean, we know a lot now about biofilms and how, organisms and biofilms can kind of, isolate themselves from like the larger milieu that they find themselves in. But I think that is, again, what I was saying earlier, context matters. Where are the microbes? How are they, sticking together? What are they making? Are they, trying to shield themselves from different things? Or are there relationships that they're having with other microbes that have other functions than they do? Like maybe there are some microbes that can do other cooler things that they like to hang out with. And so I think trying to move into, or I think I try to be really intentional in my, research in the airway space about not taking things for granted or thinking just because, the same organisms work a certain way in one part of the body doesn't mean that they may work the same way in a different part of the body. And so, I mean, hopefully that'll, lead us to some really cool things that we can discover. Because I think it's one thing to, work with microbes that you purchase from a culture collection. But I think the process for us of figuring out, what are the microbes that we're looking for? How can we, tailor this growth environment to get these kind of organisms that we're interested in? What does that look like? That's been a really exciting journey, especially for me, because a lot of the work that I've done prior has been computational. And so I think I'm very interdisciplinary in that I really like computational biology, but I think that you kind of have to do both in tandem. So it's like we kind of learn things from the data, or we can look at, patterns in like large groups of people, and then that can give us some interesting leads. And then we take up those leads and follow up on them in the lab, and then, rinse and repeat. And so I think the first step, for my lab has been trying to figure out how to move from, these are the observations that we see digitally or computationally, and how can we, work with those samples, pull out those microbes, culture those microbes in the lab setting. And so even though they're not really complicated experiments, but we around here, we get really excited about, bans on gels that we get when we're trying to identify, particular organism from a mixture, or when we get sequencing results back and it's like we've got a pure culture, like yay, like that's really exciting.
For listeners and viewers, it's important to remember that when you do microbiome studies, you can find out who's there. And as the technology improves, you might be able to find out hints about what they're doing. But in some ways, and I don't at all want to sound reductionist, it's better to actually have some of the organisms that you can manipulate. And as you say, I mean, here's a situation where if you ask an individual what percentage of microbes in nature can you culture in the laboratory on particular media, that number is actually quite small. Mm-hmm. But when you find something you can manipulate and study a little bit more, it gives you another set of powerful tools.
Yes, I think that they really, really inform each other. And I'm excited because I think now that the technology in both spaces, I think there's all kinds of really cool things we can do at the bench. There's all kinds of growth chambers and assays and, ways that we can read or watch or observe bacterial growth in real time, like that in conjunction with all of the really powerful computational tools that we have and workflows that are out there. Like I think bringing those two together has a lot of really cool potential for this, host associated microbe overall story, because we can, take some of the things that we see in the data and actually ask those questions and get some definitive, answers. Like, what are these bacteria doing? What can they make? And how do our immune systems or our immune cells respond to those things that they make? And so I'm really excited to be, working in this space where those two areas that typically are, two sides of the same coin, I think, but you can spend your whole career on one side or the other. And so I'm really intentional about trying to kind of thread the needle of doing both. So
I think that's important. And I'm an older gentleman, so the idea of being super reductionist is the way that I was originally taught. I well remember when I was reviewing papers a lot early in my career, when people were first studying biofilms, I would say, well, you're looking at one particular organism in this biofilm. How does that relate to nature? Because you're not going to generally see one type of organism in a biofilm. And then they'll say, well, we've got 2 or 3 that we're looking at. And that isn't really an answer. It's a universe, isn't it, when you think about it? But you have to start somewhere and you build on it. That's one of the really cool things about science, especially today. I think we're all looking at the news, and this idea of portraying what you do to the public is really important. What I like about your research is that it's something that people can actually appreciate and understand without diving into the microbial weeds, for lack of a better term. I really admire it. And that brings me, if I may, to some of your outreach activities.
Yeah. So so in my lab, we do, outreach events where we have been, going to give elementary school students opportunities to kind of engage with different types of experimental, science. And so we've had these science nights that we've done, in partnership with the PTOs at the school, and they put them on every year, and we just have it's almost like a bazaar type thing, but like for, science and art and math and technology. And so my lab has been able to, work on their skills in storytelling for, how to talk about these scientific concepts in a way that, kids can understand and that parents and, families can understand. And it's, flexes a very different set of muscles, I think, than what you typically are engaging with. We're like in the lab, doing science or getting ready for a conference or something like that. It's a whole different, sphere of communication. And so we are able to do a lot of cool things. We've done this for both, really young. So from, I think about age 5 to, 7, but also for like the older elementary around, 7 to 10 years old. And so we've had to, scale what kind of activities that we do. And, with the younger kids, it's sometimes it's just, really cool. Like we'll have like a volcano making station, for example, and talk about, the properties of different liquids, acids, bases, add some, color, and things explode and overflow. And that's really cool and really fun. And we have it to where we invite, other labs in here at the university to come and put up a booth or a station where they can talk about their research. We've also included the local museums, or there's a couple of biotech companies here that have had, public or communications teams that will come and do little activities. They've done, extracting DNA from strawberries. Like that's also like really cool. Like it's really popular. And so with microbiology specifically, I just find it really amazing to be able to, share this, world of things that exist that we can't really see without assistance. And that's really cool. It was one of the things that got me hooked on microbiology when I was young, which I'll come back to in a second. But for the most recent event that we did, we had a station, as you can see in the photo, where we had, interacting with microbiology with our senses. And so we had, I think it was look, smell, and touch. And so for look, we had made some E. coli expressing the green and blue fluorescent proteins and had it in a cardboard box so that it could be dark enough to where if you look in the box, you could see the bacteria glowing. And then we had I brought some of my sourdough culture for smell. And so because, it's very fragrant once it's, really alive and bubbling and everything. And so, most kids could recognize like, oh, it smells like bread, or it smells kind of like, they could recognize that it smelled like sort of something that you want to eat. And so we talked about, microbes and how they contribute to bread and fermentation and what that means for, yogurt and other kinds of fermented foods. And then for touch, we had a SCOBY from a kombucha. And so one of my students has a roommate who brews her own kombucha. And so she had a huge SCOBY, which if you don't know, it stands for symbiotic culture of bacteria and yeast. And it's like this big, like squishy, like gel matrix of yeast and bacteria. And so we put that into a box and had a hole, and kids had to like reach in and touch it and guess like, what it was. And so that the kids really liked that. And I think the parents did too, actually. They're really excited to like learn about how microbes, contribute to making these things that they either, enjoy food wise, or they could really like relate to that, which was really great. And we've also had, people and like other microbiology students come and set up viewing stations, as you can see in the other photo, where we have, microscopes with pre-made slides and plates with things that we've already kind of streaked out and preserved, and help the kids see what they look like under the microscope, and, help them focus them. And, I see you have in the background a lot of the microbe, like the stuffed giant microbes. Those are also a big hit. And so we usually lay those out too, and like, talk about the different things that, cause different kinds of illnesses. And it's been a really, really gratifying thing to be able to do, and a really good way to kind of involve the whole scientific community of our general area in doing that kind of work. And the kids seem to really enjoy it.
I've got to just applaud you, because you have no idea how things like this that you just described changed my life when I was a little kid. I didn't grow up in a very nice area. I no negativity. I was born in Compton and raised in North Long Beach. So, very low income areas. And fortunately at that time, there was a lot of support for education in the state of California. And there were these opportunities where I could actually try experiments or learn things. People actually spent time doing that. And I've noticed there's not as much of that going on. So it's wonderful that you're doing that, because it really does change things. By the way, there's an artist, and I'll send you a link if you're interested. I'll certainly put it in the show notes, lives north of me in Port Townsend, who actually makes sculptures out of the kombucha scobies.
That's cool.
And I want them.
Yeah.
I know I can't get them, but I want them.
Yeah, it's really cool. And I think that, for me, my interest in microbiology started when I was 8. My parents bought me a microscope. That was my birthday present. And the microscope came with, a couple of stains. It came with, real glass slides, which felt like super official because they were like sharp and they could actually, like cut yourself on them. And the cover slips and a little bottle of brine shrimp and like other things to make, wet mounts. And my mom, actually, this was back when malls were malls. Yeah. And we had a science supply store, or science like surplus store. And they sold like really cool, like rocks and, just, it was like a museum gift shop almost. And so they actually had, Petri dishes and, agar. And so my mom looked up, how do you make a Petri dish that we could, culture? So we got some like beef broth or chicken broth or something and made plates. And I just went around and made a culture collection of our house, essentially. And so I was swabbing the doors, the toilet seats, the toilet handles, and whatever, the raw chicken. I was just like swabbing everything and just watching the different kinds of things that grew. A lot of them did not smell pleasant.
No.
After growth, I discovered. But that was really the, impetus of me, being really captivated by this idea of like all these microbes that exist and grow places, and we can't actually see them, but they're just as much a part of our lives as the things that we can see. And that was kind of the start of that for me.
It's life-changing. And, when I say that, you don't have to go get a PhD to love science. And I do think more and more in our society, being able to show folks, I have a saying, and I don't have an elegant way of saying it. There used to be this expression, a big tent, where lots of different people with different ideas could be under the same tent.
Mm-hmm.
I would say what we need is a big petri dish where you have lots and lots of different people. I mean, can you imagine my poor parents? My father was a firefighter. My mother did like data entry. They had no idea what to do with this right here, but they supported it anyway. And I think that's the goal, to have people in your life that are able to do outreach and support things. Let them ask questions. Let them figure out new ideas. People get shut down all the time, and we've got to really, if you'll forgive me, preach that science is an important part of our lives and understanding it matters.
Yes, absolutely. And I think, especially now as I think there's a disconnect between, the process of science, or like how we do science. Like sometimes it's slow and it's very iterative, and we find out little things at a time. And our, information environment that we live in is often moving at lightning speed, where, you can ask your phone a question and your phone's going to tell you something. It may not be correct, but like you get the feeling of, like, oh, I asked and got an immediate answer. And, the process of, learning about the world we live in is a lot slower than that, and has to be done, methodically, with like lots of checking and lots of, what's the word? Corroboration, to verify things that we're seeing. And I think that there is a disconnect, or I see that like in my students, where we talk a lot about, how do you think, or how long do you think it took to, figure out these things that we take for granted now as like, this is just how it is. Like that took a long time. And a lot of the advances that we've made microbially in like public health, like those usually came off the backs of a lot of tragedy and a lot of loss. And we've been, I think, distanced from that as our society has, moved forward. But I think that's an important thing that we kind of have to remain, anchored in as we are trying to, move forward.
I do know that when I teach first year students, and we do part of the tiny earth technique, where you isolate microbes from the soil or other sources and show that they have antibacterial activity. And I will have students who have utterly no interest in what I have to teach. And that's okay. It really is. I'm not making a judgment about it. But when they see those zones of inhibition, an organism that they found, and we don't know what it is yet, but they get to find out in class, and they know that it has an effect on several different types of bacteria, which are relatives of things that cause disease, their eyes light up and they're suddenly interested. So honestly, that's where I think we need to go, to kind of start early with that, as you've been talking about, and get people to understand that, as you say, science is a process.
Yeah.
There I go preaching. So as we begin to think about closing out, what are a couple things you would like all listeners and viewers to remember about your research?
Well, one, I think it's great to always be, thinking about your airway microbes, and where the places where you are, spending your time, and what things you may be exposed to that maybe you haven't thought about. So I think that that's something that is important. And I think, as we emerge from, our pandemic, where we all kind of got a crash course in, knowing that there are things that are around us that can, have an impact. But just like thinking about, how we interact with our immediate outside environment, and what that might mean. And I always try to, especially lately, to make a pitch for funding science. Is that like my science is, I think it's really, really amazing, really cool, and has a lot of things that can offer the world. And we need money and resources to be able to do those things. And so in whatever way that people can, I would, encourage people to be involved in, championing the importance of investing in our knowledge and people that are trying to do the work to answer these questions that we have.
And it's more than simply an investment. It's an investment with known returns when you look at the record. And I'm reminded, again, because of my age, there used to be a senator named William Proxmire, and he would pick out research that he thought was silly and that would get all over the news. And it used to make me so angry because what he was doing is picking and choosing, and it would turn into a big battle, and you would have to prove yourself. And what I like is saying, let's just explain to people what the value of research really is. And that's what you've done here today.
I hope so. I try. I tried.
Ari, are you going to be coming to ASM Microbe in Los Angeles?
I will not be in Los Angeles, but I will be at ASM Microbe next year.
Well, I will see you at one of those places, and it will be nice to shake your hand.
Yes, agreed.
I really enjoyed today's episode, and I want to wish you and yours the very best.
Thank you. I really enjoyed it.
Thank you. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, as always, epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Ari Kozik can be found in the Department of Molecular, Cellular, and Developmental Biology and the Department of Internal Medicine, Division of Pulmonary and Critical Care Medicine at the University of Michigan in Ann Arbor, Michigan. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile each time. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.